All right, well, we are at 8 a.m. We will start grand rounds. Thank you all for coming. Delighted to have Dr. Tori Williams here, from our very own division of geriatrics. And to introduce Dr. Williams, we have Dr. Sanjay Asthana, our division chief of geriatrics and gerontology. Sanjay?
Yeah, thank you, Lynn. Thank you. Good morning, everyone. Thank you for joining the grand rounds today. It is a great pleasure to introduce the speaker, Dr. Victoria Williams. Dr. Williams is an assistant professor in the Division of Geriatrics and Gerontology. She's a clinical neuropsychologist by training, and she has significant expertise in the area of neuropsychology and brain imaging. And she's used that expertise over the years to examine the relationship between brain behavior in older healthy adults and those who have dementia. Dr. Williams received her PhD in clinical neuropsychology from University of Houston in Houston, Texas, and then went on to do her clinical psychology internship at UCSD and the associated VA hospital. From there, she went to Boston and did 2 postdoctoral fellowships. The first one was at the Boston Medical Center and the affiliated VA healthcare system. This fellowship was in clinical research, and then which she went on to Mass General Hospital at the Harvard Medical School and trained at the internationally renowned Interdisciplinary Brain Institute and conducted some very exciting Alzheimer's research and clinical and translational research. We brought Dr. Williams here in 2019 as an assistant professor in the Division of Geriatrics. She's very closely involved with a large NIH-funded study called the Wisconsin Longitudinal Study, and is conducting some breakthrough research using that cohort. In a presentation today, we'll have some interesting data from that study. She's very productive at this early stage of her career. She has published 32 peer-reviewed papers, and many of those papers actually have appeared in prominent journals, including neurology, Alzheimer's and dementia, human brain mapping, and so on. She has made 28 conference publications, both national and local, and she has made a number of invited presentations as well. She's funded by 2 NIH grants. She is the co-leader of the Neuropsychology Service of the Wisconsin ADRC. And as I mentioned earlier, she's a co-investigator in the WLS R1 grant and has 50% effort utilizing that cohort and doing her exciting research. She's also a co-I in the past on 3 NIH grants. She is on the editorial board of the Journal of Alzheimer's Disease. She serves as an associate editor and is a consulting editor on neuropsychology. I would like to invite her to make a presentation on environmental exposures on dementia research. Dr. Williams.
Thank you, Sanjay, so much for that kind introduction. I'm really excited to speak with you all today to share some new and quite intriguing findings that arise from the Wisconsin Longitudinal Study, where we demonstrate differential effects of mid and late life environmental respiratory exposures on late life all-cause dementia risk, and how these associations may vary as a function of ApoE genetic status. So here are my disclosures. So I'll start today's talk off at the end, which is dementia. So dementia is the primary cause of disability and dependency among older adults, with 1 in 7 Americans over the age of 71 currently living with some form of dementia. In fact, most of us listening today will be impacted by dementia at some point in our lives, whether it's developing the illness ourselves or providing care or support for a loved one struggling with the disease. And we know that dementia is actually an umbrella term, which is used to describe the clinical syndrome of progressive cognitive decline, which can arise from a variety of different underlying etiologies, such as Alzheimer's and vascular dementia being the most common. Yet what is shared across all of these different pathologies that give rise to the dementia syndrome is that we currently have no cure. Thus, perhaps the best strategy to reduce the burden of dementia in our society is to look at ways that we can perhaps prevent the disease process from occurring in the first place. So what places an individual at increased risk for dementia? Decades of research have identified a variety of factors that may either increase or decrease one's risk for developing dementia, with these exposures occurring at different points along the life course. And these include things like lifestyle choices like diet, health, exercise, sleep, and substance use, social factors such as education, relationships, stress, and income or wealth, specific exposures to things like chemicals, toxins, respiratory hazards, or even the use of prescription medications. And then more general exposures that affect populations at broader levels, such as socioeconomic disadvantage, air pollution, and factors associated with urbanicity. And together, these factors are collectively termed the exposome, and it reflects an individual's unique combination of exposures throughout their lifetime. Importantly, most of these exposures are potentially modifiable, and thus represent a possible mechanism or a point where we may intervene to possibly reduce dementia risk. However, not all risk for dementia is modifiable, and most notably, the ApoE4 gene is significantly associated with increased risk for dementia in late life. But importantly, these genetic factors are also known to modulate the impact of exposures on the phenotypic expression of dementia, where both the timing of the exposure and their interaction with the genome may vary across the life course and differentially impact dementia risk. So today's talk will specifically focus on the impact of environmental and occupational respiratory exposures, which as a just general concept, environmental and occupational exposures are known to account for over 12% of worldwide mortality, with dementia risk being included in that statistic. So as a result of this presentation, I hope that you'll be able to summarize the existing knowledge of the effects of environmental exposures on dementia risk, to describe how genetic factors like ApoE may interact with environmental exposures to modify this risk, and finally to consider the principle of genetic pleiotropy to possibly explain the differential gene by exposure effects on dementia risk that we see across the lifespan. So back in 2020, there's a high-profile paper that was published by the Lancet Commission that reported results of a comprehensive meta-analysis and synthesis of the existing literature to identify the most robust and reliable modifiable risk factors for dementia, which collectively they found were thought to modify about 40% of dementia risk. And then the authors then kind of ordered these risk factors across the lifespan to explain like when along the life course these factors may exert the most impact on dementia outcomes. And as you can see, air pollution, exposure to air pollution late life was determined to be a significant modifiable factor among older adults. However, we know that the presence of, in particular, amyloid pathology, which is a hallmark characteristic of Alzheimer's disease, it can be detected in the brain 20 to 30 years before the actual onset of the clinical dementia syndrome. And this suggests that there's a protracted or prolonged prodromal period of disease progression or accumulation of pathology that may extend well into the later portions of midlife. So thus, midlife may be a more etiologically relevant time period to address these modifiable risk factors in order to prevent the initial onset of dementia pathologies in the first place. And we know that in midlife, adults spend most of their waking hours in the workplace, as we are now. And this suggests that perhaps occupational related exposures also constitute a key source of environmental exposure within this age range. In particular, we know that both occupational respiratory exposures and air pollution may share a similar mechanistic pathway in how it may impact the brain. And as the majority of respiratory or airborne pollutants are too large to cross the blood-brain barrier to exert direct neurotoxic effects on the brain itself, the hypothesized kind of primary mechanism by which inhalants impact brain health is through mechanical inhalation, which can lead to an inflammation, and it's really the secondary inflammatory response which impacts brain outcomes. Yeah, we know that research examining the effects of these midlife respiratory exposures on specifically on dementia outcomes is quite scarce in the literature, and it really remains unclear whether these types of exposures in midlife may impact late life dementia, and whether it's on par with the effects we see in late life air pollution exposure studies, which we'll start with there. So air pollutants arise from a variety of sources in the environment. And there are 5 key air pollutants that are closely monitored and regulated by the Environmental Protection Agency due to their known adverse impacts on brain health or just general health. And these include particulate matter, nitrogen dioxide, carbon monoxide, sulfur dioxide, and ozone. And the number of studies that have been examining the effects of late life air pollution on dementia outcomes has grown exponentially since the release of the Lancet Commission paper identifying late life air pollution as a significant and potentially modifiable risk factor for dementia. And among those, by far, PM 2.5 or particulate matter is the most frequently studied air pollutant, and it also shows the most reliable and robust associations with higher levels being associated with increased risk for dementia. So this next slide, I'll just show you a synthesis of existing studies that was published in the Science of Total Environment in 2021. And I don't expect to go through all of this, but I just want to point out that all the studies looking at the effects of air pollution on a variety of cognitive outcomes are listed along the top row. And on the left column, we have the different effects or outcome measures. And so I'm just going to hone in on studies that looked at dementia as an outcome. And we can clearly see that most studies have this red indicator, which denotes a statistically significant finding of an adverse association of higher levels of air pollution leading to increased risk for dementia, which overall just suggests this overall reliable and consistent effect across studies and cohorts. And as I mentioned before, studies looking at the protracted effects of occupational exposure on dementia outcomes are a bit scarce in the literature. And here I'm just going to point out kind of two papers of interest. So the first is a meta-analysis that was published just last fall that really combined data from over 85 different studies looking at a very wide range of occupational exposures and how these may be associated with risk for vascular dementia in particular. And then a second paper reports findings from a large kind of population-based cohort from the Netherlands, which here they prospectively examine the effects of various different types of occupational exposures on risk for actually non-vascular types of dementia, so largely Alzheimer's disease. And these are results across both studies, which I won't go through in depth, but I will point out that the sorts of exposures in red boxes were associated with significantly increased risk for vascular dementia and non-vascular dementia. But what I will want to point out is that if you look at the results across these studies, that the types of occupational exposures that were associated with increased risk for both vascular and non-vascular dementia tend to have can be considered inhalants or hazards to respiratory health. So to briefly summarize what we know so far, we know that exposure to ambient air pollution and especially particulate matter is reliably associated with increased risk for dementia. And although there have been fewer studies looking at the effects of occupational exposure on dementia outcomes, studies have generally supported that there is a pattern of supported respiratory hazards as particularly increasing risk for both all-cause dementia, I should say. But there are a couple of limitations that are worth pointing out and discussing. So among the studies looking at air pollution, most of them have employed just single pollutant models where they look at one pollutant individually. But we know that air pollution metrics tend to be correlated. So for example, in urban areas, we see high levels of traffic-related emissions, so PM 2.5 and NO2, which tends to be lower in rural regions. And also these complex mixtures of air pollutants tend to be more representative of what's encountered in the natural environment. Also, most of these large studies looking at air pollution and dementia outcomes are based on kind of huge population-based cohorts. And many of these rely on really crude measures of dementia outcomes. So those that are defined algorithmically or based on kind of medical record, which in some cases may over or underestimate dementia prevalence. And then finally, most of these studies have really been restricted to older adult populations. And there are very few studies that look at the effects of kind of pollution or air pollution in midlife and how they impact late life dementia risk. Some of the limitations of the occupational exposure studies, as I mentioned before, there's really been kind of a lack of positive research in this area. And that the studies that exist, oftentimes the exposures are actually estimates that are based on reported occupational categories. So they're not a direct measure of whether an individual was actually exposed to occupational respiratory hazards. And a few of these studies, if any, I really had a hard time finding any, have really considered whether ApoE may interact with exposure to modify its effect on late life dementia outcomes. So returning back to our overall framework, so we talked about the occupational and air pollution exposures. Let's now break down a little, explore more about the ApoE gene and how this may modify this effect of exposure on dementia risk. So we know that the ApoE gene exists as 3 different isoforms, which vary in their prevalence in the general population. So ApoE3 is considered the wild type with an allele frequency of around 80%. The E2 allele is the rarest variant, whereas the E4 allele has a frequency of around 14%. And since individuals carry 2 different copies of the allele, this leads to about a quarter of the population carrying at least 1 copy of the ApoE4 variant. So although the 3 isoforms only differ from one another by a single amino acid, this minor change can produce profound differences in both the structure of the resultant ApoE protein and the amount of protein that is generated. And this leads to important differences in how these different allele types are able to modulate other key cellular processes. So in the CNS, we know that ApoE is primarily expressed by astrocytes and plays a really key role in cholesterol trafficking and metabolism. In fact, that's considered the central role of the ApoE gene in the cell. Yet aside from this primary role in lipid transport, the ApoE variants have also been shown to differentially modulate many other important cellular processes and functions, which include synapse formation and neuronal signaling, the immune and inflammatory response, antioxidant capacity and glucose metabolism, and maintenance of the blood-brain barrier. However, the ApoE4 gene is by far most known for its role in modifying the risk for Alzheimer's disease, where carriage of the E4 allele is considered the most potent genetic risk factor for sporadic and late onset Alzheimer's disease. So compared to ApoE3 carriers, those carrying ApoE4 allele have a 30 to 35% increased lifetime risk for developing Alzheimer's dementia, whereas the E2 allele has been shown to perhaps offer some protection against dementia risk. So in the setting of Alzheimer's disease, which is characterized by the presence of amyloid plaques in the brain, the ApoE4 allele has been shown to interact with many of the mediators that are believed to be involved in Alzheimer's disease onset and progression. And these include things like exacerbating neuroinflammation in Alzheimer's disease, increasing synaptic dysfunction, as well as contributing to greater metabolic dysregulation. Importantly, the E4 allele has also been shown to significantly modify amyloid deposition. Some work by our own Toby Bethauser in our division has very eloquently showed that ApoE4 carriers tend to have an earlier age of amyloid onset, and that the difference and then those E4 carriers also show a faster rate of clinical progression to the dementia syndrome. And the E4 allele has also been shown to modulate the accumulation of tau pathology as well. So thus, the ApoE4 gene can be linked to several Alzheimer's disease-associated pathways. But what about its ability to modify the effects of environmental exposures on dementia outcomes? And so to explore that, we turn to some existing studies in this area. So the first I'll describe is data coming from the Women's Health Initiative Memory Study, or the WIM study, which is a cohort of older adults. Older females. So this is an all-female study. And they examined whether the effects of late life particulate matter exposure was able to be linked to risk for all-cause dementia. And their results suggest that indeed E4 carriers, so those either hetero or homozygous E4 carriers, tend to show an increased risk for dementia based on level of air pollution exposure, while the effective E3 carriers did not reach a level of significance. And similar findings have been shown, or a similar pattern of findings showing a preferential risk among ApoE4 carriers to the effects of pollution on dementia risk have also been shown in other large cohort studies, including the Solia cohort. And this is a community-based sample of also older women, but this one is in Germany, as well as the Vietnam-era twin study of aging, which is a cohort largely comprised of older male veterans. So taken together, these studies suggest that there is a reliable effect of the ApoE4 gene in modulating the risk of environmental exposure on dementia outcomes, with ApoE4 carriers showing being the most sensitive or showing the greatest risk in relation to air pollution exposure. And interestingly, the initial study in the WIMS cohort I mentioned earlier had a complementary sub-study that they included in their article that also looked at the effects of late life particulate matter exposure on the amount of amyloid deposition using a transgenic mouse model. And the mouse there were mice that were either exposed to control ambient air or 24-hour cyclic particulate matter exposure. And results suggest that those exposed to the particulate matter condition or the air pollution arm of the study showed almost over double the amount of amyloid deposition compared to the AD mouse model and control air, suggesting that there is a strong link between air pollution and the onset of dementia type pathologies. Okay, so returning back to our overall framework, we'll just finish building out our main aims of this study. And these are to examine the effects of midlife occupational exposures to respiratory hazards and late life exposure to air pollution on all-cause dementia risk, and whether these effects of life course exposures on dementia outcome may differ by ApoE4 carrier status. And in order to adequately model these life course kind of time-dependent sorts of effects, we must rely on a complete longitudinal sample that could accommodate this task. And here we turn to the renowned Wisconsin longitudinal study, which will form the population where we'll be kind of looking at these effects. So the Wisconsin longitudinal study is a random selection of 1/3 of all 1957 Wisconsin high school graduates. And this is a true random selection. And the initial sample included over 10,000 recent high school graduates, with a select sibling of each graduate that was added around 1977, which brought the total cohort up to around close to 20,000 participants at one point. So the Wisconsin longitudinal study is predominantly white, non-Hispanic individuals, and this is representative of the demographic of high school graduates in the state of Wisconsin back in the 1950s. And although it has limited racial diversity, there are adequate diversity in other areas. So for example, roughly 30% of the sample are still residing in rural areas, and 20% of the sample were living below the poverty line in 1957. And this study offers robust life course data collection beginning at age 18 and covers nearly every facet of participants' lives. So early surveys really focused on educational, occupational outcomes, but as the cohort aged, the focus of research shifted on collecting more health-related data. And now that the sample is generally in their early 80s, the Wisconsin longitudinal study has partnered with the Wisconsin Alzheimer's Disease Research Center, led by Dr. Asthana, where we're now going back out into the field and trying to determine the dementia prevalence of the sample. And so using this life course framework, this is kind of how we're going to situate our model, where the primary dependent variable, or what we're interested in the most, is all-cause dementia outcome, which is determined around the age of 81 for the graduates. With air pollution exposure also being determined a bit earlier in late life, so around the age of 72. And then occupational exposure is based on data that was collected during the working years, so between the ages of 54 and kind of retirement age. And so I'll go through each of these primary variables, and we'll go over kind of how they were defined. So we'll start with dementia outcome, and our protocol for assessing dementia prevalence in this sample is very explained in detail in a previous publication if you're interested, but I'll just give you a very brief overview of kind of how we went about this. So it was a two-phased approach where the initial living and reachable sample was given a phone screen to identify risk for dementia based on scores on a cognitive phone-based screening measure, the TICS. And about a quarter of the participants that we were able to reach failed the TICS, so placing them at increased risk for cognitive decline or dementia. And so these participants were selected for a more detailed phase 2 full assessment, where we did cognitive testing, functional assessment, and a thorough medicational examination. And this information was then presented and discussed in a multidisciplinary case conference to arrive at kind of a clinician-based diagnosis of dementia. And from that process, we identified about 197 cases of dementia. The rest of the cases that failed the TICS, half were about MCI, and then half were found to actually be performing with cognition in the normal range. And then there were quite a few cases that either had died or that were unable to participate in the testing for whatever reason. And so we did collect some proxy information using the dementia questionnaire, which was able to identify another 296 participants with likely suspected dementia. And so when we combine those numbers, the overall prevalence of dementia that we were able to detect in the sample was right around 10%. with 493 individuals meeting criteria for research-based dementia diagnosis. And the remaining just over 5,000 were found to not have overt dementia. And if we break down these two dementia groups by frequency of ApoE allele carriage, we find results that we may expect given the risk of ApoE4 on dementia outcomes. So in our dementia group, about 43% of that sample were carriers of the ApoE4 gene, whereas only 22% in the non-dementia sample were ApoE4 carriers. So to define our next variable of interest, we'll focus on occupational exposure, this was based on a response to a survey question asked both at midlife and at retirement age about whether they were exposed to dangerous conditions in their work and the types of hazardous conditions. And we selected cases that reported exposure to hazardous chemicals or other hazards to respiratory functions. And of this group, about 20% reported a history of this type of exposure. And most of the individuals with this exposure reported the frequency of almost always to frequently. So late life air pollution exposure was based on the participants' residence or primary residence in late life in 2011, where we linked their primary residence to EPA monitored air pollution data that was averaged across that year, centered upon when data was collected. And because pollutants are known to co-vary kind of based on population density, as I mentioned before, we were interested to see whether there are any clear patterns of how we can kind of reduce or simplify these 5 pollutant measures into a composite. And as you can see, there's the PM2.5, NO2, and CO tend to show a clear elevation in more urban areas. So this graph below looks at population density with the urban areas and most densely populated regions on the left, kind of going down to less populated or rural regions on the right. Whereas ozone and SO2 tended to show the lowest levels in more urban areas, but then were elevated in the most rural areas. And when we looked at the actual covariance of these 5 pollutants using a factor analysis, we identified 2 factors which mapped quite well onto these urban and rural patterns, such that one factor we termed the urban pollution exposure factor, and the other constituted rural pollution. And these 2 factor scores were what we ended up using in our full model. And so it's also important to think about other things that may geospatially co-vary with air pollution exposure. And one of those, we considered other confounders, such as area deprivation index. So this is a composite measure representing neighborhood level disadvantage. And we define that for each individual at the corresponding time point of our air pollution exposure. We similarly controlled for occupation-based indicators of socioeconomic status, which corresponded to their job at the time of the exposure in midlife. And so to continue building out our full model, we also included and controlled for adolescent premorbid IQ. So these are IQ scores obtained in high school, lifetime, their age, their lifetime education level. We controlled for comprehensive smoking index, which is basically, it is a composite measure of both frequency and duration of smoking across the lifespan, as well as lifetime disease burden of cancer, hypertension, stroke, diabetes, and heart disease. We also included the fixed effects of participants with known ApoE4 or ApoE genotyping, as well as sex. And so based on complete case data, we were able to find a little over 3,800 participants that had complete data for all of these included variables. But to also account for missing data, we ended up using a weighted regression model. So our primary outcome in this study was dementia outcome, and that was defined as all-cause dementia. And we used binary weighted logistic regression to look at primarily the effects of occupational exposure and air pollution exposure, consisting of urban and rural late life air pollution, and how these may differ as a function of ApoE4 status. Okay. So here are findings, a summary of our key findings from the study, where the effect size here reported as odds ratios for all-cause dementia, which are broken down by midlife occupational exposure and late life urban and rural patterns of air pollution exposures. And then within each exposure category, we show the independent effects for ApoE4 carriers and non-carriers, as well as the marginal effect, which is the average of the two and is considered the main effect of the model. And so the effect estimates highlighted in red indicate that the results reached a level of statistical significance. And here we find that in late life, we see the air pollution exposure was only associated with dementia risk among ApoE4 carriers, in particular, high levels of urban air pollution. Oh, sorry, I missed that. Here we see that midlife actually increased risk for occupational exposures on dementia risk were associated with ApoE4 non-carriers with an odds ratio of 1.62. Whereas in late life, associations with urban and rural air pollution were only associated with ApoE4 carriers. So higher levels of urban pollution increased risk for dementia in late life among ApoE4 carriers. Whereas we had an unexpected finding where actually higher levels of rural air pollution exposure decreased risk or was protective against dementia risk, again, among ApoE4 carriers. And given this unexpected finding, we decided to do a follow-up sensitivity analysis where we repeated the regressions, but we rotated through each of the individual air pollutant metrics to kind of see if there's anything in particular that was driving this unexpected effect of rural air pollution showing a protective effect against dementia. So here are results from that sensitivity analysis. And the graph here is broken down by ApoE4 carriers and non-carriers. And the reason why there's 5 estimates for occupational exposure is that it corresponds to each individual air pollutant model in order of how they appear in the table. So we can clearly see here, again, there's that isolated effect among ApoE4 carriers that higher exposure to occupational respiratory hazards significantly increases risk for dementia, which we don't see in the ApoE4 carriers. Yet we see the opposite pattern with regard to the single air pollutants where, so there's the occupational exposure, where ozone actually appears to be driving the protective effects that we see with the rural pollution exposures. And then particulate matter was driving the adverse effects on dementia outcomes, which is fairly consistent with the studies that I've described earlier. So this leaves us with 2 kind of main findings to really unpack a bit. So one is we find that ApoE4 modifies the effect of exposure on dementia risk differentially across the lifespan. And 2, that ozone exposure appears somewhat protective against dementia risk in late life ApoE4 carriers. So we'll start with our first effect, which is the fact that we see a differential effect of pollutants across the lifespan that are modified by ApoE4 status. And so on ApoE4 non-carriers, we do see that there's an increased risk for dementia related to occupational exposures, but we don't see a significant effect related to late life pollution. And then we see this opposite effect among ApoE4 carriers, where they seem to be protected against the effects of midlife occupational exposures, but are more sensitive to late life air pollution exposures. And so perhaps the best way to kind of explain these differential effects on the timing of air pollution exposure on dementia risk is to really revisit the mechanisms by which respiratory exposures may most readily impact the brain and whether this process stands to be linked to some of the ApoE associated functions. And so indeed, we see actually that inflammation likely constitutes a shared pathway by which airborne pollutants stand to impact the brain and is differentially modified by ApoE allele variants, such that the ApoE4 allele is associated with more of a heightened and pro-inflammatory response. And yet to fully comprehend our findings of how ApoE may differentially modulate this effect of respiratory exposures based on timing across the lifespan, it may be helpful to look at a couple studies that consider the ApoE response to inflammatory stimuli in both healthy young adult states compared to how ApoE may function in the context of aging and chronic inflammation. And so to start, I'll just briefly go over a study by Gale et al., who recruited healthy young adults who were given an LPS or lipopolysaccharide injection. And this is an acute inflammatory stimuli that initiates a immediate inflammatory response. And their primary outcomes were looking at how ApoE moderated the LPS response to temperature, as well as to the production of inflammatory cytokines. And so here are results from that study, which I'll walk you through. So first, I'll just point out that the E4 carriers are denoted in the open circles, and the E3 carriers or non-ApoE4 carriers are denoted in the filled circles. And we see that there is a elevated effect of LPS on temperature among ApoE4 carriers, where they show a heightened temperature response to this inflammatory stimuli. ApoE4 carriers also show a quite significantly increased response in TNF alpha or tumor necrosis factor alpha, which is an acute inflammatory cytokine. And then also ApoE4 carriers tend to show an earlier onset of interleukin-6, which again is a pro, but also an anti-inflammatory cytokine as well. So taken together, this may suggest that perhaps in a healthy state, we know that the inflammatory response actually serves a normal physiological function to protect cells from disease and to promote healing. And so it is perhaps the case that the ApoE4 heightened response in a state of healthy or homeostatic state could be a protective mechanism. But what about in the setting of advanced aging? How does the ApoE modify the inflammatory response there? And this study is a little bit different, but it's a cohort study that uses data or participants from the Framingham Heart Study that identified a proportion of those participants in mid to early life who were free from dementia, who had monitored C-reactive protein levels extracted from blood at various time points across the life course. And so CRP or C-reactive protein is a kind of just general measure of inflammation. And the researching team took these serial measures of CRP to define kind of groups reflecting high to low levels of kind of chronic inflammation across the adult lifespan. And we're interested to know whether these inflammatory profiles were associated with risk for Alzheimer's disease and whether ApoE was able to modify that association. And so here are primary results from that study. And again, I'll walk you through them. So on the y-axis, we have the cumulative incidence of AD. So of the people that had these inflammatory markers, how many went on later to develop Alzheimer's disease in later life? And on the x-axis, we have the group broken down by kind of levels of chronic inflammation, where those on the left had the lowest levels of chronic inflammation, whereas the group on the far right had by far the highest levels of chronic inflammation. And these inflammatory groups were further parsed by ApoE4 or by ApoE status. And so there's just a couple key results to point out here in this graph. One is that among ApoE3 carriers, chronic inflammation did not seem to impact the risk of later developing Alzheimer's disease. But regardless of whether you had low or high inflammation, you had about the same cumulative incidence of Alzheimer's in that group. Whereas among ApoE4 carriers, there was a very clear and quite pronounced increase in risk for Alzheimer's disease developing later based on your level of chronic inflammation kind of in midlife and the point leading up to the disease. Whereas those with the highest levels of chronic inflammation were almost showed twice the incidence of Alzheimer's disease compared to ApoE4 carriers with low levels of inflammation. And so this may suggest that perhaps in a chronic inflammatory state, the ApoE4 response may be deleterious and actually kind of increase risk for adverse outcomes. So when taken together, these studies suggest that in kind of a homeostatic state, the inflammatory response may perhaps serve a vital defense function of maybe removing injurious stimuli, initiating healing, thus offering protection. But in the setting of kind of chronic inflammation, which can be seen with advanced aging, this pro-inflammatory properties of the ApoE4 allele may shift from a protective to more of an antagonistic role, to thereby exacerbate kind of neurodegenerative processes and the pathological onset and progression of dementia. And so the theory of pleiotropy may serve as a framework to kind of interpret these opposing effects of ApoE across the life course. So in particular, antagonistic pleiotropies suggest that a single gene may show differential effects across the life course by promoting survival and fitness early in life, but then shifting towards this more detrimental effect or adverse effect on survival and fitness in late life. And so although this effect can simply just be attributed to perhaps a loss of protective function, so whatever was the gene was protecting the organism in early life, and then that just is lost over time. And there's also kind of a sub-hypothesis of that this effect really is more of a gain of toxic function, where the allele is actually protective across most of the life course until there's some type of external event or stimuli that then shifts it over to a gain of toxic function. Um, so, for example, the ApoE4 inflammatory response may enhance normal physiology in a young kind of healthy state, but may shift towards one that promotes pathology and is antagonistic in late life. And ApoE has previously been proposed as a possible pleiotropic gene. So the most straightforward example of this is that ApoE4 carriers tend to show the highest levels of fertility and enhanced reproductive fitness early in life. So thus this ApoE could serve to enhance survival. Whereas as we've clearly seen in late life, carrying the ApoE4 gene significantly increases risk for dementia and disease. But perhaps representing a more nuanced interpretation of this antagonistic pleiotropy effect, ApoE4 allele carriers also show protection from differential protection from a variety of adverse environmental exposures in early to midlife compared to non-carriers. Although the direction of this effect or protective effect then shifts in late life, where ApoE4 carriers tend to show more adverse effects of environmental exposure on dementia risk. And so there have been a series of studies that have looked at different types of exposures across the life course and how these may vary by ApoE4 status. And so I'll just briefly go through a couple of these as an example. So we know that ApoE4 protects against the adverse effects of lead exposure on 24-month-old cognitive development, so it's protective. In utero particulate matter exposure was associated with larger adolescent brain volumes, but this effect was only seen among ApoE4 carriers. So again, suggesting perhaps a protective response to exposure. Smoking, this has been mixed, but there was a recent meta-analysis of over 37 studies that suggest that smoking increases risk for dementia, but this is preferentially among ApoE4 non-carriers, which is kind of akin to what we find with our respiratory exposure in midlife in our study. And then in late life, this shifts towards being a deleterious effect. So higher late life air pollution levels is increased with dementia, higher dementia risk, but preferentially among ApoE4 carriers, which is again what we see in our study. So returning kind of to the main findings and to tie that together, once again, results from this study suggest that in midlife, the ApoE4 allele may be protective against these adverse effects of occupational exposures on dementia risk. Possibly attributed to the pro-inflammatory tone among E4 carriers, which may serve to kind of mitigate these pollutant-associated inflammatory effects among ApoE4 carriers earlier in life. Yet in late life, perhaps in the setting of maybe chronic age-related inflammation, or perhaps due to another undefined trigger, exposure to even low levels of environmental hazards, such as air pollution levels in this study, may exacerbate the risk for dementia among ApoE4 carriers, but show minimal effect in older ApoE4 non-carriers. So taken together, the ApoE4 mediated inflammatory response kind of may provide this plausible framework to interpret these time-dependent exposure effects on all-cause dementia risk. And this really places inflammation right in the intersection of environmental exposures, ApoE function, and mechanisms that are associated with dementia risk. However, it's really important to also note that this study was observational in nature, and it does not include any direct measures of inflammatory markers by which to kind of directly test these interpretations of findings. And it's also important to recognize that there are other potential mechanisms. So for example, exposure-related insults to vascular functioning that may likely contribute or even better explain these key findings in our study. Also, it's important to remember that the nature and source of occupational exposures were not explicitly defined in the WLS sample. And so it very well could be that direct chemical neurotoxic effects from especially midlife occupational exposures may actually account for this increased risk of all-cause dementia in this study among our ApoE4 carriers. With again, the inflammatory response may offer protection among E4 carriers. So overall, it's my hope that this hypothesized framework centered on inflammatory mechanisms generally serves to inspire future research in this area, with the understanding that we still have a long way to go to fully understand this intricate and highly complex system that promotes dementia and Alzheimer's disease types pathologies in the brain, and how these environmental or external exposures impact that pathway. So the second, we'll turn our attention now to our other main finding. which is basically the protective effects of ozone exposure on dementia risk among ApoE4 carriers. And although this was quite unexpected, there have been few studies that have actually looked at the effects of ozone on dementia outcomes. And most of these have reported deleterious or null effects, but very few, if any, have actually stratified by ApoE4 status, which may explain this finding or lack of findings. But what I did find that was interesting was kind of a recent vein of research that has identified ozone as actually a naturally bioactive molecule that offers antioxidant properties and could be a potential new strategy for in aging and neurodegenerative disorders. And this review identified several key areas which controlled ozone exposure may actually benefit cellular functioning. And I'll just point out that the activation of anti-inflammatory systems was listed as one of the key ones. And these same authors published a companion paper that same year, kind of delving deeper into these mechanisms. And interestingly, the anti-inflammatory effects of ozone were really attributed to a decrease in TNF-alpha and interleukin-6, which if we go back to that study looking at the acute inflammatory response in ApoE4 carriers, these are the exact two cytokines that showed significant differences among the ApoE4 groups. So taken together, it's possible then that ozone is actually kind of counteracting the adverse inflammatory response in late life, particularly among ApoE4 carriers. So to wrap it all up, this study showed that in midlife, occupational exposures to respiratory hazards were associated with increased risk for dementia, but only among ApoE4 non-carriers. Whereas increased risk for dementia among ApoE4 carriers was only found in response to late life elevations in air pollution, and in particular, particulate matter. This suggests that there is perhaps the ApoE gene is able to differentially modify the effects of respiratory exposures on dementia risk across the lifespan, possibly supporting an antagonistic pleiotropy such that the ApoE4 is actually protective until there's some type of gain of toxic function due to some external stimuli, which could possibly even represent just chronic inflammation that accumulates over the age span, where the ApoE4 pro-inflammatory response shifts from one that promotes physiology and survival in early life, but then becomes antagonistic, promoting pathology and senescence and aging as people get older. And that these inflammatory responses really do tend to tie things together. We also found that late life ozone exposure among ApoE4 carriers was actually protective against dementia risk. And although this was a quite unexpected finding and certainly deserves replication in future studies, it is of interest that O3 is gaining ozone is gaining attention for some of its anti-inflammatory properties, which may actually kind of dampen the exacerbated or may be beneficial among ApoE4 carriers. And so I'll just leave you with a couple of ideas for future directions. So in this talk, we, like I said, focused a lot on the inflammatory pathways and how that might impact dementia risk. But it's important to know that there are many other pathways that could be possible explanations for these sort of differential life course findings. And that although our study only considered all-cause dementia risk, future work should really try to break this down by suspected dementia etiology to see if there's differential effects of mid versus late life on vascular or non-vascular dementias. A step towards this aim is that we were recently awarded a renewal grant in the WLS study, where we are now going back into the field and collecting blood samples from all living WLS participants, where we then hope to extract levels of amyloid in the blood-based samples to look at that are determined to be biomarkers of Alzheimer's disease in the brain. This work also should highlight that future research should really consider these complex complex differential associations between exposures and how these may be modified by the genome and affecting particularly cognitive outcomes. And that these effects may actually differ across the life course. And so focusing on late life or even in midlife, you may be missing the whole picture. There's also a range of other different exposures that you could apply to this same sort of model. In fact, that is something that we plan on doing in future studies. And then finally, given the limited racial diversity in the Wisconsin longitudinal study, these results should really be replicated in more racially or ethnically diverse samples. We know that the strength of the association between ApoE and Alzheimer's disease varies as a function of race and ethnicity. So just verifying that these findings are reliable and robust and applicable to the more general population would be extremely important. And then I'll just end by acknowledging the key investigators in this study. So Dr. Sanjay Asthana and Pam Hurd, Dr. Michael Engelman and Sterling Johnson, as well as our wonderful staff that are on this study. So we have nurse practitioners, Anne and Brittany, our wonderful program manager, Kate, our new scientist, Ralph, and we have Camille. And they've all been instrumental in making this work possible. I also want to thank the Wisconsin Survey Center for their diligent help with data collection, and the Wisconsin longitudinal team who manages this data set and curates all of this wonderful work that we're able to do. And then finally, and most importantly, I'd really like to extend an appreciation to this large cohort of participants in the Wisconsin longitudinal study that have contributed more than 67 years of data to make this sort of work possible. And with that, I will end, and I would be happy to take any questions.
Great. Thank you for that wonderful tour de force, particularly trying to tease out the different contributions over a lifetime. I give you huge kudos. Maybe we can stop sharing now.
Sure.
And a couple of questions that have come up. Maybe start off with the question is, what actually is the ApoE protein and what does it do?
Sure, that's a great question. So the ApoE4 protein are expressed typically on astrocytes in the cell, and their primary role are to kind of bind lipids and cholesterols in the brain and to transport them across different membranes and maintaining kind of cholesterol homeostasis in the cell. And I know I kind of went over a bunch of different ApoE associated functions as well, including things like systemic inflammation and but really it's the way that these ApoE proteins express themselves that either particularly in the case of inflammation, the ApoE4 allele, sometimes the amount of protein that's generated is what modulates these sort of responses. And so higher levels of ApoE4 or ApoE proteins that are generated by the like E2 or E3 carriers has a differential effect on things like the inflammatory response compared to kind of lower levels of ApoE proteins generated by ApoE4 carriers. And then further, the actual proteins themselves that are produced by the different alleles have different affinity for binding and can have more kind of nuanced ways in which they impact these other diverse arrays of pathological functions and processes in the cell.
Great. And a question from Carrie Gleason. So she has some recollection of hearing that an ApoE4 carrier may actually have advantages in some learning in early cognition. Are you aware?
Yes. This talk got way too busy, as I'm sure you saw, but I left out a lot of really important facts. So yeah, there's an entire hypothesis looking at the kind of antagonistic pleiotropic effect of the ApoE4 allele just based on its effects on cognition. I didn't talk about that because cognition is not really a mechanism, it's more of the expression. But yes, early ApoE4 carriers show actually much higher performances, particularly on measures of memory and other sorts of cognitive domains, and that these effects tend to decline, so that ApoE4 carriers actually show a faster rate of decline compared to non-carriers. And the protective effects of ApoE4 on cognition, again, like according with this kind of antagonistic hypothesis, flip in late life and then are associated with faster rates of cognitive decline and worse cognitive outcomes.
Okay. Then from Bob Probelski. So do you see us using ozone therapeutically in selected patients with the appropriate phenotype?
It's possible. And, research of these large population-based studies have actually shown that there is this gradation and like rural versus urban effects on dementia risks. And, perhaps it is, the ozone effects. I mean, that's wild and out there. But, I do think this was an unexpected finding in our study. I was surprised and somewhat pleased to see that like it's been considered as a possible therapeutic approach for neurodegenerative diseases. My husband's a chemist, and when I told him about O3, or he's a geologist, and when I told him about O3, he was like, "Oh yes, that's a very bioactive molecule." And so it was kind of an interesting literature to delve into just for a little bit and to see how people are thinking outside the box and developing new ways that we may be able to kind of modify the dementia phenotype. But I will say that from this brief review, there are certain studies that are, or maybe even in clinical applications, where ozone actually is being used as a therapeutic agent in some instances. So maybe it could be extended to dementia.
Yeah, more of a precision medicine approach, really,
Yes.
with the right genotype. I'm going to interject my own question. So globally, biomass exposure is, the major cause of pollution and exposure. Do we know anything about indoor biomass exposure and the risk of Alzheimer's?
When you say indoor biomass, is that like indoor pollutants?
Yeah, like wood stoves and
Oh, yes, yes.
cooking stoves. Yes.
Yeah, there are studies that have looked at both indoor and outdoor sources of pollutants. I think the mechanisms would stand to be the same. That, the same types of PM 2.5 that you would see related to traffic emission would be also the same sorts of particulate matters that may affect you from kind of wood burning. But yes, there have been studies that have shown that both indoor and outdoor sources of pollution or respiratory pollutants do increase risk for dementia in late life.
And then, do you have any information or know anything about the role of various nutritional supplements and how they interact with the ApoE phenotypes and
Yeah, that's a really interesting question. And on it, after kind of, thinking about how inflammation really tends to unite some of these key concepts, it made me kind of extend that to think about some of the other protective and risk factors that have been identified. And certainly nutrition and Mediterranean diet are one of those. And that's not really hasn't been my area of study, but I do think that the effects of our dietary choices also impact kind of our levels of chronic inflammation. I think that this concept of like the inflammatory response and where it goes wrong being a central kind of driver of Alzheimer's disease is gaining traction in the field. So it would be exciting to kind of extend that to basically explain some of these other mechanisms in the exposome.
Great. And Nizar had my question, or related question, since it's all inhalational injury and of course everything goes through the lung, are you thinking about stratifying patients on lung disease? He mentioned asthma. I was actually thinking of COPD or the number of COPD exacerbations.
Yeah.
Because we know both of those diseases are increased in many of the exposures that you mentioned as well.
Yeah, and that's fascinating. I don't know if I don't think off the top of my head that this study has any type of objective measure of like respiratory function or capacity. But I will point out that throughout my literature search, ApoE4 doesn't just modify disease risk for Alzheimer's disease, but actually COPD is one of the other diseases that ApoE4 is able to, moderate as well. And so that would be a really interesting question to further stratify and look at kind of, and I think in the case of COPD, I think ApoE4 carriers have differences in their baseline kind of lung capacity. Again, not my area. I apologize if I didn't quite eloquently state that, but yes, I do think it would be kind of interesting to look at the actual respiratory function. I think in this talk, I was trying to think of ways that would directly tie it to the brain. So the secondary inflammatory response is kind of the most accepted mechanism by which most inhaled pollutants can exert influence on brain health. But yeah, those are.
And like the COPD exacerbations are also chronic inflammation, same mechanism. So
yeah, exactly.
I'm going to ask another question. Have you or has anybody looked at acute the role of acute exposures versus you're looking at lifetime, but I'm thinking post wildfire or World Trade Center or volcano eruptions and
Right.
track those patients, those
Yes.
people over time. And
Yeah, and we actually were just in Wisconsin exposed to one of those effects. I don't, was it a year or two ago where there was those Canadian wildfires that were
oh yeah.
horrible air pollution days. Yeah, we haven't looked at that in this study, and I honestly am not quite sure. I mean, I assume somebody may have done this in the literature already, but it would be really interesting to kind of look at these, you know, mass exposing environmental events and seeing how that may impact trajectories of later life associated risks or disease.
Yeah, and I know many of the pulmonary folks monitor them over time, and it'll be interesting to see if you can do some initial cognition or
Well, I love to
follow-up over.
collaborate. So if anyone over in pulmonology wants to send me an email, I would welcome that. You know, I'm a neuropsychologist, and so I did my best here with these mechanisms, and I do find it really fun to kind of go through and try to find these, you know, ties that could link everything all together, almost from a bird's eye view that someone still has when they're not, you know, deep in the weeds.
Yeah.
But you know, I'm always willing to learn and explore new exciting mechanisms or alternative hypotheses. So I would love to hear from anyone in the audience who have ideas.
Great. Well, there's a couple more questions, but we're out of time, so I'm sure Tori would be delighted to take by email additional questions. Thank you so much, and appreciate all of the work that's gone into it, and look forward to more analysis of an amazing cohort. Really.
Yes, it is amazing. So thank you so much for this opportunity.